This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 725, recorded on February 26, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 38 Fahrenheit, which is 3 Celsius, and it's sunny. And our snow is in piles on our grass, but gone from the streets and sidewalks.
Mm-hmm. Yeah, it's a little warm today. It's above freezing for once, and the snow's melting. Also joining us from Austin, Texas, Rich Condit.
Howdy. 53 degrees and cloudy, so meh. It's going to be 62. It's meh for the next couple of days. I can handle it. I'll be okay.
From Western Massachusetts, Alan Dove.
Good to be here. It's clear blue skies, 40 Fahrenheit, 5 Celsius, and I think we're just moving out of winter and toward mud season now here in Massachusetts.
Mud season. We have 2 guests for you today. We are trying to span the United States. Our first is returning for her second time on TWiV. She was with us way back on 228 from the University of California, Berkeley. Eva Harris, welcome back.
Thank you. And I just looked it up. We're 64 degrees here in Berkeley, California. Really clear skies. I have a lovely view here. So
nice.
Thank you.
Yeah, I was out there, and we had you and Britt Glounsinger on a TWiV. And I remember it very well because, you know, at the end I asked you, I think, who would you would have dinner with if it could be anyone? And you said, you got to get back to me. I don't know. And in fact, you emailed me a few days later. You couldn't come up with something during the episode. I thought that was very funny.
We did one on Zika, if you remember. I think that was at the Boston meeting.
Yes, then we had a chat, you and I, at the Boston meeting, which I think was a celebration of the opening of the needle.
Right. Yep.
And that we talked about Zika in Nicaragua, right? That was very good also.
It was just blowing up
at
that moment. Remember that, yeah.
Also joining us for the first time from the University of Michigan, Janet Smith. Welcome.
Thank you. Thank you, Vincent. It's a pleasure to be here. And Kathy and I are in the same place.
You are.
So it's also 38 degrees here, and it is beautifully sunny out, and the snow is rapidly melting.
So you think this is the last snow for northern parts of the U.S.? No? More?
No.
I wouldn't go
but
so
it's
far
almost
as to say that.
the end. It's close.
So March can get snow, right?
Yeah.
Yeah, but it'll be that horrible East Coast snow if it snows in March. It'll be wet and heavy.
So Janet's not been on TWiV, but we talked about your work on TWiV 465 with Theodora Hatzianu, ApoBec C3H structure. Remember that?
ApoBec 3H bound to RNA. Yes,
bound to
of course
RNA.
I do. Yep.
Yeah, some surprises there. Yeah, that was really cool.
Big surprises. That's right.
Yeah, and I got to meet and talk to Janet's student, Jen Bone, in preparation for that. So that was fun.
Cool. You had a leg up on us, huh, Kathy?
Well, only a day before.
She usually does.
Before we start,
a step ahead.
let's hear a little bit of your histories. Eva, you told us a long time ago, but we have all new listeners now. So tell us again about where you're from and your training.
Well, of course, my path is like this, but very circuitous. So I did my undergraduate in biochemical sciences at Harvard, and then I did my PhD in molecular and cell biology at UC Berkeley, actually, and kind of a postdoc in molecular parasitology. But while I was doing all of that, I was also setting up a program in Nicaragua and many Latin American countries. It was kind of a forerunner many, many years ago of what became global health. And so I've kind of had this dual career, if you wish, that has a big part of capacity building in developing countries, but then has kind of grown into a very strong field component of our arboviral work. And so I've been able to kind of marry those two parts so that my laboratory here at UC Berkeley, where I am in the Division of Infectious Diseases in the School of Public Health, does work on molecular virology, pathogenesis, and human immunology, while our partner in Nicaragua, we have a number of studies that we talked about before, actually, on hospital studies, cohort studies, and directly in communities for evidence-based approaches to controlling arboviral diseases.
And if I remember, I think you're originally from New York City, right?
Yes, I grew up in New York City, actually, and Paris, kind of back and forth before my compass moved south.
Paris and New York, not bad.
Nice.
Not a
So
bad.
your association with Nicaragua, did that grow up just professionally, or is there other things that drew you to that?
Other things that drew me to that, it was essentially while I was doing my undergrad, actually, I just have this deep love of biology and of how the cell works, the molecular and cellular mechanisms. But I also felt a deep drive to kind of do something in the real world and couldn't figure out how to marry those. And they kind of come together over these 30 years now. But at that point, there was a revolution going on there, and I wanted to make biology part of it. And that's kind of what grew into this big, long program, which kind of was a forerunner of kind of global health and bringing tools in country to make a difference from the bottom up.
Janet, where are you from?
So I'm also an East Coaster. I am from Philadelphia. And I did an undergraduate degree in chemistry at a state college in Pennsylvania, which is called Indiana. And I then
IUP, yeah.
Indiana
University of Pennsylvania.
Yes, Yep. And so I then it got way more exciting when I studied biochemistry, when we got sort of towards the end. And then I decided biochemistry was a thing for me. So I went to the University of Wisconsin in the biochem department and did my PhD there. And that's when I started doing, well, it wasn't called structural biology then. And I was actually doing small molecule crystallography. I did structures of antibiotics that inhibit protein synthesis, but I really, really wanted to work on proteins. So as a postdoc, I worked with your colleague, Vincent, Wayne Hendrickson. And I was at Columbia, actually with him before he moved there, and then for a few years at Columbia. And have, sort of fell in love with proteins and have been working on protein structures ever since. And I took my first faculty position at Purdue University, and I was there for 18 years. And I am not a virologist, but I sort of learned about viruses by osmosis from attending seminars, many of which were by the people who work on virus structure and worked on virus structure at Purdue, and the collaborators they invited for seminar visits. And every seminar starts out with a life cycle slide. So I sort of got a little bit, starting with non-enveloped viruses that could actually be crystallized, like rhino and so forth, all the way up to more complicated enveloped ones. But I stayed away from it when I was a junior faculty member, just to establish a little bit of independence. And eventually, in the early 2000s, began collaborating with a virologist, Richard Kuhn, on non-structural proteins. And around not too long after that, I was recruited to the University of Michigan into the Life Sciences Institute here. And the NS1 project was one that had been a sort of thorn in our side, because we and many, many other labs around the world couldn't figure out how to make the protein. And once I got here, in a core facility that I established, we hired a really good expression expert, and he just kept at it and, would periodically say to me, this project is not going to beat me. I am going to figure this out. And it took a few years. And it was one of those things where in retrospect, you say, oh, yes, of course, it all makes sense. But ahead of time, you couldn't have figured it out. So that's how we got here. And otherwise, in my life, I am actually more known in the structural biology community as a person who has facilitated the access to synchrotron radiation for people in my field. And I'm the scientific director of some NIH-supported beamlines at the Argon synchrotron. That takes a goodly chunk of time.
When were you here at Columbia?
I was in Columbia in the early 80s.
We might have overlapped. I came here in 82.
We did.
Yeah. I never...
So where's... Pardon my ignorance, but where's the Argon synchrotron?
It's outside Chicago.
Chicago.
Okay.
Yeah.
It's about 20 minutes drive from O'Hare on a good day.
I was in New York too at that time.
You were.
For listeners who are not structural biologists, or biologists, the synchrotron is there aren't very many of these, and this is a resource that's critical for the types of structural biology that
it's an X-ray. It's an X-ray source, and it's many orders of magnitude stronger than what we can produce in a lab.
And many millions
Department
of
of Energy.
dollars.
There's one at the really best one in the country is at Argonne. That's my opinion. There's one in Berkeley. There's one at Stanford. There's one at Brookhaven. And there's one at Cornell. And there's a little teeny one in Louisiana.
But everybody who wants to do protein crystallography needs to find access.
Yeah, yes. Something like 90% of the structures that now end up in the database are done with synchrotron. It's absolutely essential.
It's like telescopes for astronomy.
Right.
Yep.
And we're talking about a machine that takes up a warehouse.
Yeah.
Right.
More than. Yep.
You mentioned Indiana University of Pennsylvania. So I learned of it about, 5 years ago. I got an email from a professor there, and he told me how he used TWiV in his biology course. He sent me this. He said he has students pick an episode, listen to it, then write a summary and answer questions. And at the end, he wrote, by listening, you will see how scientists integrate and talk about information. It's just great. It's just great. So good science.
And he's right.
I was very excited to get that. It was one of the first inklings that we had some effect on education, right? which I really like. I think that's important. All right. So today you're both here because you have published together a wonderful paper. And I have to say, after reading this, I wanted to thank you both because it's so well written and it's so clear and it makes sense. And I understand every word. And this is not the case for many papers.
Especially papers in a journal like Science, where you have this much space to explain this vast amount of it's extremely well written and concise
and very dense. Of course, there are single sentences which are probably many experiments all condensed into one sentence.
It takes a while to read, but it's all clear. You just
I think it's great. It is structural basis for antibody inhibition of flavivirus NS1 triggered endothelial dysfunction. We have two co-first authors, Scott Beering and David Akey, and we have two last authors, Janet and Eva. And I do recognize Stephen Elledge and Michael Minna. And after reading the paper, I recognized what they did also. So we'll get to that. But let's start, perhaps, Eva, you can tell us about NS1 and flaviviruses. What do we need to know to understand this paper?
You better start with flavivirus because, a lot of our crowd now is tuning in expecting coronavirus stuff. So this
still exists, yes.
Yeah.
So, and are very important. In fact, these flaviviruses are arboviruses, arthropod-borne viruses, and the flavies are transmitted by mosquitoes, especially Aedes aegypti and Aedes albopictus, and actually about half of the world's population is at risk for infection. And there's 50 million and above 50 million cases per per year and 100 million infections. So it's a big deal for dengue, which is what I have been working on the longest. But there's also very well-known Zika virus, which is a close cousin and actually interacts immunologically. We've actually just shown as well is able to enhance following dengue infection. So there's like all this interaction between flaviviruses. And then we have yellow fever, which is another systemic flaviviruses. And then there's the neurotropic flaviviruses, such as West Nile, which people are probably familiar with in this country, Japanese encephalitis, St. Louis encephalitis, and then Zika actually is kind of in between. It's both the systemic and neurotropic. And so I've been studying dengue for a very long time because there's so many questions around it. There's 4 serotypes, imaginatively named 1, 2, 3, and 4. And as I mentioned, they kind of each have a flavor, but they all cause overall a similar clinical syndrome. But they can both, one infection can both protect or enhance a subsequent infection. So there's a lot of really interesting work around the human immunology, which we've done a lot of work both in the lab and in our cohort studies and such in Nicaragua. But there's also, many leads. One of the biggest issues with dengue in particular is this vascular leakage clinical manifestation, which will lead to shock and then death. So it's a small percentage of the vast numbers of people who are infected who will develop this more severe form of disease, which used to be called dengue hemorrhagic fever and dengue shock syndrome. In 2009, WHO reclassified to dengue without or with warning signs and severe dengue. But fundamentally, the most kind of distinctive pathophysiology is this vascular leakage. And the kind of dogma has been, and is still true, that we think that there's an immunopathogenesis. Clearly, like as I just mentioned, one serotype followed by another can lead to what ends up being this kind of exaggerated but misdirected immune response, which eventually kind of hand-wavingly leads to vasoactive cytokines through activation of immune cells, and then to this endothelial permeability and vascular leak. So in 2000, maybe 13, 14, and we published this in 2015, we stumbled on the fact that NS1 by itself, which is the only secreted protein of the flaviviruses, can actually cause vascular leak all by itself. So this was kind of a paradigm shifter, because it wasn't only this immunopathogenesis, but rather actually a viral protein, which could then directly cause the hallmark feature. And we actually stumbled upon it, in parallel to a group in Australia, Paul Young's group, and we published together with back-to-back in Science Translational Medicine at that point, kind of taking, and what was interesting there is that we had kind of found 2 different mechanisms that NS1 could do in terms of vascular leakage. NS1 is, again, it's the only secreted protein of the flaviviruses, and it has multiple other roles in viral replication, which had been known both genetically indication, but then also had been worked up by various groups, and then also has a role in immune evasion and being able to evade complement, and had been implicated in pathogenesis via complement, actually, but not any role for the protein itself. And so we and Paul Young's group and others have since kind of developed this theme of NS1 as a viral toxin. And so what Paul's group had shown and others is that the NS1 can actually trigger immune cells to release vasoactive cytokines, in addition to the virus being able to do that. And then what we took on was the endothelial cell intrinsic or specific activities of NS1. And what we've shown is that NS1 can lead to the destruction of the endothelial glycocalyx, which is a kind of a brush border on the endothelium of proteoglycans, which are a physical barrier to fluids and solutes to avoid vascular leak. And we've shown by multiple different pathways are activated. As it activates sialidases to cleave sialic acid at the surface, activates cathepsin L, which then activates heparinase, which then cleaves heparin sulfates and syndecan at the cell surface, and ends up destroying this glycocalyx so that the solutes and the fluids can be right up against the endothelium, the endothelial cell cells. And then we've also shown that there's tight junctions which hold these together, tight junctions and adherent junctions, and that NS1 has a separate role in mislocalizing the tight junction and adherent junction proteins, which is another whole story. And then we also actually have another story potentially with inflammasome activation. So this protein can do tons of stuff around vascular leak. And so that's kind of an introduction to, and what we showed is that not only is it dengue virus, but we then expanded, and that's the paper that you discussed actually, Vincent, in an earlier TWiV, but where we discovered kind of serendipitously also that NS1 proteins from 5 different flaviviruses. We looked at dengue, West Nile, Japanese encephalitis, Zika, and yellow fever. We're actually able to cause endothelial permeability and actually vascular leak in mice all by itself in the human endothelial cells from different tissues and in the actual organs in mice that reflected the disease tropism of each of these 5 different flaviviral diseases. And so we think that it actually has to do with viral dissemination and allowing virus to get across endothelial barriers inside the organism to be able to replicate in target cells and such. Right. So that's kind of where we left off. And then I can, eventually tell you about why we stumbled onto the, collaboration with Janet and what that led us to.
You do mention one thing in the introduction that if you can immunize mice with this protein, it will protect them, right?
Yes. So that's actually how we came across it. We were working on various vaccine strategies, essentially that we had NS1, because of this problem with ADE, antibody-dependent enhancement, and the ability of the structural proteins on a flavivirus like dengue, meaning the E protein and PRM, to be able to cause antibodies that will then subsequently enhance another infection. NS1 was a really nice target because it's a non-structural protein. It could potentially serve as a vaccine antigen. But at that point, all of the studies that had been mice, it was in mice, that had been challenges were all neurotropic models of mice. And generally, dengue is not a neurotropic disease. So since we had, because of our work, had developed mouse models that had a systemic viral vascular leak as the symptom of mortality, we then thought, let's look at NS1 in its capacity to protect. And actually, to our surprise, just NS1, even though it's a non-structural protein, was able to protect 100% against our lethal challenge of vascular leak. And then we thought, maybe NS1 is actually involved in pathogenesis in causing that leak, and thereby the vaccine being able to prevent it. And then we developed a toxicity model in the mice, and then showed that it caused leak in mice, and then it went from there.
And this notion that a vaccine against the structural proteins could cause antibody-dependent enhancement, that's not theoretical, right? I mean, that's something that has actually played out in vaccine development.
Many people have predicted that, and including our studies, which showed at the same time that in children in Nicaragua, a certain range of antibody could cause enhancement of a subsequent dengue. We were concerned with the Sanofi vaccine at that time because there was unequal protection by serotype and also unequal protection, even in the pre-phase 3 trials, but in naive versus dengue immune recipients. And so there was a lot of concern that, in fact, the antibodies might hit this danger zone. And in fact, we think they did, because there is more severe disease and more hospitalizations in children who are naive, who are then vaccinated. And so that was, yes, it's not theoretical.
Right. So
go ahead.
Is NS1 essential for virus replication? Can you knock it out? And what's the phenotype of knocking it out?
It's essential for viral replication. And it's in the viral replication complex. And
okay, that's right. All right.
Like S4B inside. So there's interactions in the replication.
Okay.
When you immunize mice with NS1, do they get infected but not develop disease? Does virus still reproduce in them?
Yeah, well, the thing, it's a little complicated in the sense that to actually work with the viral, at least dengue, you need so the interferon is very, very effective in clearing flaviviruses. And so dengue and other flavors have developed immune evasion strategies to get around the human interferon system. But those strategies, which is as with STAT2 and various other, there's a number of different mechanisms, essentially do not work in mice. And therefore, mice don't get systemically infected very well with, unless you're in an interferon knockout model. And so, so you end up having to work in these models. Nicely with NS1, we can work in only wild type mice because we're looking only at the protein itself.
Okay.
So essentially, it's not fully protected against infection. It's more against this more severe disease.
Got it. Okay. Now in this paper, and a key part is the monoclonal. So you made that, I think, before you started this collaboration, right? How was that made? And what did you select for?
We were this was made in like the year 2000 or 2001 by Robert Beatty in our lab. And actually we have this great, he's a professor actually in molecular cell biology here at Berkeley and teaches immunology and is also kind of a member of our lab. And so we developed a system when he has a class that makes monoclonal antibodies every year. Mm-hmm. We like slip in our proteins. The class makes antibodies and then they start and then we finish the cloning of the hybridomas. And so at that point we thought, let's make some NS1 and NS5. And so we just had this little factory going and we had these great proteins and we have a wonderful NS3 antibody actually also that many people use in the field that were made by the students. And so we had this NS1 antibody that we used for many different purposes. And then got very interested in it. We actually had made a number of ELISAs to be used in kind of non-proprietary manners for diagnosis because NS1 is a great because it's a secreted protein. It follows the viremia and you have an antigenemia, and so it's a good diagnostic marker. And so we had been using these antibodies and kind of worked them up. And then started to look for ELISAs to be used in the field. And then we had noticed. We started looking at where does it bind? How does it, and then we came up with this. We have a very strong 2B7 monoclonal that was great. And then when we started working with Janet, we thought, wouldn't it be cool to see where it bound? Because we actually already had data that we thought indicated that 2B7 bound to the wing domain, because we knew that the wing domain, there's 3 domains of the NS1 protein. It was involved in binding in kind of our pathogenesis work. We had noticed that. And then this and 2B7 block binding. And we thought, we had some evidence. We thought that it bound the wing. And then we said, let's just lock this down with Janet. And so we centered the protein and lo and behold, it bound to the beta ladder. But what's really cool about the paper is that we actually can explain mechanistically how it blocks different pathogenic functions of NS1, but can sterically hinder, from, even though it binds to one domain, it can actually sterically hinder a different domain. And we'll get to that, but I want to pass it over to Janet she can talk a little bit more about the structural kind of finding.
That was great.
Yeah. I want to jump in because I think that I hope that Janet's going to tell us a little of the background about the structure of just NS1 itself, and that would be why it's rational for you to center the antibody.
So the story in the cell is that NS1 is the genome is translated, it's plus sense RNA translated into a polyprotein, and it has a bunch of signal sequences in it, and so that get various bits inserted through the ER membrane. And NS1 is the only non-structural protein that ends up on the lumen side of the ER membrane. And somehow or other in that over there, it is essential for replication, as Eva said, which happens in these, there's also a lot of remodeling of the ER membrane. So there's this little place in vagination where all the enzymes are and the RNA is made and all the proteins are made and stuff happens. But NS1 is over here on the other side of the membrane and it has to be there. And there's very nice data, indirect, but genetic data that some part near the N-terminus of NS1 interacts with a little membrane protein called NS4B, which is essential for replication. And it kind of builds this replication complex at the ER membrane, where all the work is taking place on the inside. And here's NS1 out here, and it's associated with the membrane. So what we knew when we started the structural work was that it, by cross-linking, it looks like it's a dimer when it's in the cell. It gets secreted, as Eva said, and it's out there, it's secreted, and where is a hexamer? By cross-linking, looks like a hexamer. So much to my surprise, those results actually turned out to be correct. Cross-linking is a little bit vague, but that turned out to be correct. So the trick was this protein loves membranes. And so and we were making it it's also glycosylated. It has disulfides. I mean, as a secreted protein, none of this is surprising. So we chose to make it in insect cells where the glycosylation would be limited to high mannose. It'd be very homogeneous, so more likely to be crystallized. Insect cells, mosquitoes, insects don't secrete much NS1 like we do if we're infected. And so most of it was retained inside the cell, but it was disulfide bridged, it was glycosylated, but it would always look like it was associated with the membrane fraction because it loves membranes. And so the expression specialist here, Clay Brown, a research scientist who works here at Michigan, is the one who figured out, tiny amounts of detergent that don't actually bust up the membrane will just kind of lift it off. You take your membrane fraction and you just kind of lift it off, and then you purify it like a normal protein. And that's what we did. And that really worked very, very well. And it's a very nicely behaved soluble protein once you get it out. And so we got crystals and
I want to just jump in because I remember I have the advantage of having heard Jen present some of this at a seminar in November, and Clay tested at least 72 different conditions of ways to get the protein expressed. And then those 72 conditions and 2 temperatures and I think 2 cell types. So it's 72 times 2 times 2.
2 cell lines, 3 secretion signals, 5 viral sources of NS1 and a couple temperatures. Yes. And then he found the best one, and then he had to figure out how it was going to, excuse me, Alan?
This fits a general pattern among people who work in crystallography who I've encountered. They don't seem to be easily deterred, right? they latch onto it, right? I am going to crystallize this. And in his case, it was, I am going to express this.
I am going to, this protein is not going to beat me. That's what he kept telling me. Anyway, so meanwhile, another lab, had discovered, so this was Felix Ray and Marie Flamont at the Pasteur, had discovered that the secreted NS1, which everyone thought, which they agreed was a hexamer, actually it contains lipid. It's this proteolipid particle out there. So we knew that, and they published in a very nice PNAS paper while we were in the middle of trying to figure out how to purify the protein. And to be honest, I was overjoyed when I saw this paper because I knew Felix is a really skilled structural biologist. And I thought, oh, good, he gave up. He wouldn't have published this super low resolution EM reconstruction. Maybe we have a chance. And so a couple of years later, we published the initial structure. So the other part was that the structure was solved. David Akey, who was one of the co-first authors on this current paper, was the first author on the initial structure determination. And he used a method actually that was developed or made to work for modern proteins and problems in Wayne Hendrickson's lab, using the sulfurs, the special scattering properties of sulfur in a protein to solve the structure. And that was quite challenging. And he did a beautiful job. So it's a lovely structure. And it was super satisfying from a structural biology perspective because it was totally consistent with what all of the bits of biochemistry and biology that were out there about the protein. So it has a strongly hydrophobic surface. Okay, good. It likes membranes. We got, and it's a dimer. It's like a dyed-in-the-wool dimer. we solved a West Nile dimer structure, and then we had West Nile and dengue serotype 2 in hexamer form. With really, those were pretty lousy crystals, but we could see what the hexamer looked like. And sure enough, consistent with Felix and Marie's results, the hexamer was 3 dimers together with those hydrophobic surfaces facing in. We didn't have any lipid in there. We just had a little bit of detergent, but no lipid. But it was, everything fit. And then this genetic data from Mike Diamond indicating who's important for this NS4B interaction that probably has something to do with the replication function. Well, that's right next to the hydrophobic surface. So everything kind of clicked. And it's a super cool structure. And so this
Janet,
was all.
Jan, I want to jump in too, because I remember you saying that because there was this is a completely new structure.
Yes.
There was nothing to build this on. And so using that sulfur method was really important.
Right.
And then you got to name the domains.
We got to name, of course we got to name. Yeah. Well, so, yes. As a structural biologist, I never expect to find a new protein fold anymore because there are too many proteins we already know about. But this one, no matter how it's very highly conserved among the flaviviruses, but it looks like nothing else. People keep getting confused. There are many viruses that have proteins called NS1. This is like bad nomenclature. And only the flavivirus NS1 proteins are like this, and they're very highly conserved, usually 50% sequence identity or more. But it looks like nothing. It looks like nothing in anywhere else. And so this was, I think, a real problem for some of the early work is you couldn't figure out, are there domains? Are there pieces we can truncate? Or, there's nothing to build a hypothesis on. It's sort of similar to this other thing. What's the function of that protein? Maybe we can test that here. It's just zero. And so it was helpful to have the structure. And then we pretty quickly after we saw, so this is the initial work was a collaboration with Richard Kuhn at Purdue. And Richard said, oh, you really need to meet Eva. And you should talk to her. They're doing some very interesting work. And so he arranged, and Eva and I have actually never met in person. I visited Berkeley once, and you were, yes, I had a meeting with your group. You were on the phone from LAX.
I remember that. Really? Oh my God, I feel like every day I feel like.
I know, yeah. If I ran into you in LAX, I would recognize you. I've seen you a lot. But not even before the pandemic, not in person.
Wow.
So, and we had already started making domains. It was pretty clear that the protein was multifunctional and that, there are many people, especially Eva's lab, but many labs have antibodies, monoclonals. And so it's like, okay, we have a bunch of functions, we have a bunch of domains, we have a bunch of antibodies. All these things could get together and tell us which part of the protein is responsible for which function. And that's what led directly to the work. So we made some domains and sent to Eva's lab. We sent the, insect produced protein, which actually in all your assays behaves exactly like what comes out of mammalian, is secreted from mammalian cells. So then they sent us hybridoma cells for this 2B7 antibody for the latest paper, the only paper we have together so far. And then we made the Fab fragment, purified the antibody, made the Fab fragment, and then got on with the structural biology, and then got the monoclonal sequenced, of course, so that we could do decent structural biology.
Could I ask, Eva, there are some experiments with the monoclonal in this paper that are functional and important before we get to the structure. Maybe you could describe those.
Yeah, certainly. And I also do want to mention also, I didn't quite finish answering your question. So about just whether there's viremia, in the presence of vaccination and whatnot. I mean, so what we had, that's kind of part of that viral dissemination point that I had made previously. And so we had initially shown that vaccination actually reduces viral load in target organs. Although, and now we're just repeated that with the 2B7 antibody as well. And kind of the flip of that is that we've actually done some experiments, and you add NS1, you actually looks like it trends to have increases of, of viremia, of essentially viral load in organs. And actually, interestingly, Mike Diamond's group has some, like, from a completely different angle, some work with West Nile, and West Nile NS1 that seems to support that same conclusion. But yeah, so what we did is that we had looked first just to see about protection. And we have two different in vivo assays. One is systemic, as I mentioned, just a viral challenge, a lethal viral challenge, and we were able to show that the 2B7 actually could protect a different dose in a dose-dependent fashion, up to like 50, 60, 70% of animals. We also had made a single-chain FV, so a single-chain variant that didn't lack the FC portion, and we showed that that also was capable of protecting in mice. Which is important because it shows that it's the antigenic aspect, which is more important than the FC effector functions, which could also play a role, but it's not like some of the other work with NS1 directed and monoclonal antibodies that show that the FC is critical for function. But in this particular antibody, it's really, where it's targeting, which is really important. And so we have the systemic model. We also have what we call our back intradermal system, where we may have it's because we're trying to look for leak. We can just inject NS1 intradermally into the back of a mouse, a shaved back of a mouse, and we can we also come in with infrared dextran, and we can look at the amount of the pixels that the injection, whether it's just PBS, which does not cause leak, and then NS1, which causes leak, and then we add an antibody or inhibitors of pathways that we've identified, and we can see that the leak no longer happens. So it's like directly looking at dermal vascular leak, in vivo. So both of those were reversed. There was protection by the monoclonal antibody, and then we were interested in seeing what aspects of our whole cascade, the pathways that we had identified as being part of the pathogenic pathway. And we showed that the 2B7 blocks both binding, which is, obviously the first step. Although interestingly, there's kind of two parts of binding, as a hexamer and as a dimer. And when we get to the structure, we can tell you how actually both of those structures are antagonized by this single antibody. And then we looked at the ability, as I mentioned, to trigger the cathepsin L pathway, and that leads to heparinase, and then degradation of heparin on the cell surface and heparin sulfate on the cell surface. And we showed that all of that was antagonized by the 2B7, as well as the sialidase and activation to cleave sialic acid on the cell surface. So that all kind of was prevented in actually dengue, and with the 2B7. But I mean, we might get to this later, but actually there's a lot of conservation in the contact site. And so another part of the paper is looking at how cross-reactive this 2B7 is across many different flaviviruses. And so we were able to show that there's both the interaction of NS1 from multiple flaviviruses with 2B7. And then we actually showed function for Zika and West Nile, where also 2B7 antagonized the binding and the endothelial, permeability that was triggered, by the NS1. And I should just say that the kind of fundamental functional assay that we use is what's called transendothelial electrical resistance. And this is where one makes a monolayer of the human endothelial cells. And in this case, we're looking at pulmonary cells or brain, for instance, depending on the flaviviral NS1 that we look at. And then we can essentially such a strong barrier that you can measure a difference in resistance from the apical to the basolateral side with electrodes. And that's called TEER, or transendothelial electrical resistance. And so a drop in the resistance is actually an increase in permeability. And so that was reversed, when we use the 2B7.
I think that is an amazing set of activities for an antibody you just picked up by ELISA, basically.
Right.
right?
Yeah. And what was really cool about it was that when we made the mutations, so Marcus Wong, a graduate student in the lab, then made mutations in the contact sites, essentially working very closely with Janet's group to identify which ones. And then we then were not only able to quality control, essentially show that, yes, in fact, those mutations block interaction with 2B7, but we were also able to look at because they were in the beta ladder domain, and then we also had mutations in the wing domain, we were able to use that to address the question of what are the different domains of NS1 in terms of their function in pathogenesis. So another piece of the paper, which was not directly related to 2B7, but rather the mutations that we had in hand because of identifying 2B7, we were able to look at, again, that same series of functional assays and with mutations in different parts of the NS1. And we were able to show that the wing domain, we had mutations which were involved in binding, but the mutations in the beta ladder domain, which is where the NS1, the 2B7 interacts, did not affect binding, but they did affect the downstream ability to essentially cause leak, but also, the downstream elements. And so we were able to separate the domains. And we actually had a previous mutation which implicated that there are separable factors between the binding and what we've shown to be critical is the internalization of NS1 in order to trigger the pathogenic cascade.
So I think, Janet, it's a good time to start seeing what is a wing and what is a ladder, right?
Yeah, I think so too. I think Eva and I have this well set in our minds. So my background is an NS1 dimer with the orange and yellow, or dark yellow and light yellow, and then the two Fab fragments bound at the ends. And this is a cell. Sorry. With the lawn of glycocalyx.
It's permeabilizing your hand.
Yes. And then in the background, you can see a hexamer, which is further away, so it looks smaller. And then on the other side is an illustration that shows that you can have a 2B7 bound to each end of the hexamer, but you can't bind 6 of them because they get in each other's way. So let me show you a little better. So here's a 2B7 dimer. Looks like this. One is yellow and one is kind of whatever that other color is, tan. The epitope, sorry, this is NS1, not 2B7. The 2B7 epitope is here and here. If we look at this perpendicular, you'll see that it's really a cross-shaped flat molecule. Okay, and so here's an end and an end.
Are those the wings?
These are the wings.
Okay.
That's a wing and that's a wing. And this beta sheet that you see running crosswise, that's called a beta ladder.
That's the ladder, right.
And the antibody 2B7 binds at the ends of the beta ladder. Okay. Eva's group expected that it was going to bind at the wing. So there was this moment of panic when we solved the structure and had a meeting with them and told them what the result was. And they said, "Oh no, did we send you the wrong hybridoma?" And it was quickly, the answer was no. It was the right one. Anyway, so what I want you to, so here is the epitope on one monomer and on the other monomer. And then pay attention to this part down here at the bottom. If I turn 90, this is the hydrophobic surface of NS1. And here's one wing domain and here's the other wing domain. And this loop here, which in your previous TWiV you highlighted, this is what you called the wing. This is like residues 100 to 125 or 30. This is hydrophobic, and the surface is hydrophobic. And that's what we actually saw residual density for a little bit of detergent in the initial structure that we solved. And this is the amino terminus, this bit here, this beta structure here is the amino terminus. That's called a beta roll because it looks like a roll and it's all beta and it's excruciatingly dimeric. When we got the structures like, no, there's never going to be a monomer of this protein. So I'll come back to looking at it this way. So now you know this is all the hydrophobic bit. Here's the epitope. If I turn on the Fab fragments, you can see how just having the antibody bound is just going to be in the way of this greasy bit interacting with the cell.
Right.
And
so for people who are listening to this, if you took a ladder and set it down on the floor and you attach 2 boards to the sides, those are your wings sticking out, and then the antibodies are sticking to the wingtips, basically, right?
No. The antibodies are sticking. I'm going to turn this 90 degrees. The antibodies are sticking to the end of the ladder.
Oh, the ends of the ladder. Sorry.
The ends of the ladder, and the wings are out here.
Sorry, I got turned around. So the antibodies.
See, it's very easy to get turned around.
The antibodies stick to the ends of the ladder, but they stick down
protrude
toward where the hydrophobic stuff is supposed to be happening. So then the wings and everything that would be binding to the membrane can't get there because the antibodies are stuck.
They can't. And Eva and I think other groups have shown, let me turn off the antibody for a minute, that these, I'm trying to move it very slowly so that people who aren't me don't get lost. All right, now we've got the hydrophobic surface down. Right. This bit of we talked about this, WWG motif, highly conserved in the flaviviruses, is out here. Mm-hmm. And there is at least one more aromatic amino acid out there. And there's a lot of greasy stuff in here. There's a phi. Richard Kuhn calls this the greasy finger. This is the surface of the beta roll, which has a bunch of hydrophobic amino acids on it. So that really is what and presumably this is what keeps it associated with the ER membrane as a dimer when it's inside the cell. And then, I hate to say it, but magic happens. And somehow or other, you get 3 of them coming together. Let me find the threefold. It might take me. There we go. 3 of them come together. And
so the beta roll,
this is all the greasy stuff in here.
Okay.
Right.
This is all the greasy bit. And there's lipid in there when it's circulating in a patient.
It's almost like it's trying to form a micelle or something.
Well, yeah. But the contacts between the dimers are really pretty tenuous, you can see. And I think we and others have tried to crystallize this as secreted, and I think it's too heterogeneous. There's, this is not uniform lipid. We've tried to reconstitute. We have not had a lot of luck with that.
But the cool thing also is that we were with this conundrum of is it the wing or the beta ladder? And then not only did we make sure that we had sent in fact the right hybridoma, which we had, but interestingly, that's where Michael Minna had come in, because we were collaborating with him on a bunch of other antibodies and work actually with Nicaragua. And we were like, hey, can you just do a phage display system, which he had set up very nicely for a number for dengue actually, and then Zika. And we had a different project, and we just said, oh, hey, here's this 2B7. Can you do your phage display system to identify the epitope? Because there's this very nice system of essentially alanine immunogenesis across the entire genome. And so what he found also was that the beta ladder was what was targeted. So we actually had these 2 very different approaches, which led us to that. And then thinking about it more, we realized, the kind of when you saw the way it kind of drips over to the side and will sterically hinder the ability of the dimer to interact with the cell surface. But we actually believe that the hexamer is what's secreted, and that's interacting with the glycocalyx actually via glycans initially, and we have some evidence that potentially the heparin cyclase and other elements that can actually shift the thermodynamic stability such that the hexamer falls apart and opens up the tummy, the hydrophobic tummy, to interact with the cell membrane. But in the meantime, what about that hexamer? And then when we looked at the way the dimers come together, the beta ladder from one dimer is directly opposed to the wing of the other dimer. And therefore, you could just even see with the Fab that it being it's so attached to one beta ladder was sterically hindering the wing of the other dimer, even in a hexamer formation.
Yeah.
So there were like these two ways that this one antibody could sterically block both the hexamer and the dimer from interacting with the cell surface.
Right.
Which is really
right. So even though it's binding to the ladder, it's inhibiting both the ladder and the wing interactions.
Yeah, exactly.
On two different structures. So both the hexamer and the and on the dimer.
So you started off by saying that the intracellular form was dimeric and the extracellular form was hexameric.
Right, but then when it binds back to the cell, it's in a dimeric format.
Okay, so even in its extracellular mode, it can act as a dimer
when it hits the cell.
Well, when it hits, it has to find something hydrophobic.
And so and you've said it's a multifunctional protein, so it's probably doing something different in its hexameric form and its extracellular dimeric form and its intracellular dimeric form. Are those all different things?
It's definitely doing something when it's in the cell than when it's outside the cell.
Right.
Yeah.
Yeah. So in all of the structures, so we've solved, West Nile, Zika, and dengue. And then there are a couple other not from different viruses, all of the same viruses, but at any rate, in all the crystal structures, on the outside, what would be the outside of the hexamer, there are bound. This protein likes when it's purified. It's much happier if you have a little bit of sulfate in the buffer. So there are sulfate binding sites on the outside, this outer surface, and David Akey, who's the one who did all the crystallography on this in my lab, has this notion of the hexamer. He said it's like a beach ball, and it's rolling around on the glycocalyx, and it's kind of sticking to heparin sulfate, and that's helping it. And it's dynamic. It's loose. We can't make a prep that we know is, this is a dimer. This is a hexamer. Unless you use detergent, you can't do that. It's too dynamic. But I think that if it's a beach ball is rolling around, if it has a chance to get near the membrane, down through that grass, it's going to come apart and interact with the membrane. And then all of this stuff can happen. To get in, it has to get inside the cell for all of these other functions that Eva's lab has identified. But there's a lot to do.
In replication, this thing, NS1, is on one side of the membrane, and all of the other replication proteins are on the other side.
Right.
And yet this thing has an impact on replication. Is that correct?
Yes. So here's my idea.
Okay.
Is that, so the genome is organized. NS1 means it's, if you just label the things, it's the first non-structural protein in this encoded in the RNA that comes right after the envelope.
I'm looking at the cartoon that's on Viral Zone, and it does a pretty good job of this. Go ahead.
So it takes many, many, many copies of the capsid protein, which binds the RNA, and the envelope protein to make a new virus particle. And the last protein encoded on that genome is the RDRP, is the replicase. You don't need a whole heck of a lot of those. You don't need like 360 of them per virus particle. So I think there's a boatload of NS1 produced because there's no iris or there's nothing there to start translation. And so I think it may be involved in remodeling the ER membrane. Okay. I don't really know. But there could be things like that that are important for replication. But
so you're envisioning a sort of an indirect effect on replication rather than
I don't know.
Sorry,
I don't know.
And at the pathogenesis,
maybe among other things. Who knows?
Okay.
So Eva, this monoclonal will interfere with which other? It was made from dengue NS1, right? Which other will it interfere with?
Yeah, so actually a whole bunch, so we've actually shown very nice among all 4 viral serotypes of dengue. And then Zika, St. Louis encephalitis, West Nile, JEV, Usutu, Wessels-Von, and the ones that are lower are Powassan, yellow fever, and TB, but others like a whole sled. And I just want to also shout out to interesting. We are back to back with another paper, really interestingly, also from Paul Young and Dan Watterson and his group. Because again, we were chatting, we're like, oh my goodness. And it turns out that they had a similar but different monoclonal that also we figured out would probably be, binding to the same-ish place. And they also have found that this works not only, in terms of kind of the functions, but also in several different mouse models of Zika and West Nile as well. And so our paper went much more into the mechanism, and they took it more into the mouse model, but it shows kind of proof of concept that you can have a single monoclonal that can actually work across multiple flaviviruses.
Now, when you immunize your mice with NS1 and they generate their own polyclonal response against it, are they protected from other flaviviruses?
So we have not done other flavies in our dengue NS1 immunization models, but they used not immunization, but they used their antibody as like et cetera, for looking at different, because each model is different with the flaviviruses. So we haven't taken it out, although we are proposing to do so.
Yeah, I mean that would be, because I mean what you're getting at here is ultimately maybe a dengue vaccine or a Zika vaccine or what have you, and wouldn't it be awesome if you could stimulate a pan-flavy immune response?
And for therapeutics, and that's kind of more the direction that they follow.
I wanted to ask you about therapeutics. If someone is in shock, right, you could give them a monoclonal perhaps and curtail it. Is that something you're thinking about?
Yeah, I mean, it depends on the timing and getting people early enough and et cetera. But that's kind of more the direction that the other paper went, and we kind of went more towards the, kind of more proof of concept, and then showing that the functions, the pathogenic functions that we had already, identified actually work with West Nile and Zika. And I know we're up at the time, but I do want to shout out also, because just, thank you very much for saying it was such a clearly written paper, but a lot goes to Scott Beering, who was the lead in my lab for putting that together. And he's been incredible in kind of harnessing our team. A lot of this work was started by Henry Portoguardo, who did set up our whole vascular kind of leak, and many others along the way, Chun-Ling and Diego and Dustin and Francie and Nick and Robert. And so all the people, it's kind of a giant team all working on different aspects and then connecting with Janet's entire team. So it was just a really lovely collaboration.
Janet, how similar are the structures of the various NS1s that you've solved?
They're identical. You just cut off an amino acid and put a different side chain on there. They are very, very, very similar. But an antibody is going to recognize. So we can engineer it to work with yellow fever, which it's lousy with. But there's a really good yellow fever vaccine.
Yeah.
So we've probably touched on some of this at least. Is it fair to say that the NS1s from all the different flaviviruses have a role in vascular leakage or permeability that impacts on pathogenesis? Is that a fair statement? Because the different flaviviruses ultimately have different pathogenesis. Some are neurotropic, some are hepatotropic. Okay, so I'm wondering how you put all that together. Is this have to do with just enhancing spread that ultimately is manifested in other fashions because of other factors that affect the tropism of the virus?
Yeah, well, with dengue, where leak is the hallmark, I think that's, and there's multiple ways that dengue causes leak, which is why I think leak is the hallmark, so just NS1 itself has 3 different ways that we've identified already that can cause vascular leak directly, and indirectly. And there's the virus. And so it's kind of like, all roads leading to Rome of vascular leak, in this case. And so I think that's, and NS1 from dengue is the one that's secreted to the greatest degree. And it's less secreted in West Nile and in Zika, but we clearly can show that brain, for instance, microvascular endothelial cells, respond to West Nile and Japanese encephalitis, and actually Zika NS1 in terms of human cells with endothelial permeability, and like liver cells with yellow fever and dengue. And, so it kind of, it matches up. And so, and there's I shouldn't talk for him, but like I said, Michael Diamond has some evidence from a different way that, and West Nile, the levels of West Nile NS1 actually, relate to permeability and such. So we really, and so we think that it has to do with pathogenesis and viral dissemination through, and we have our own data with dengue as well that shows exactly the same thing. So meaning that there's, this getting across endothelial barriers inside the organism, we think is, really mediated or facilitated, I should say, by this protein. And therefore antibodies and vaccination that include it, are all the more able to block different aspects of pathogenesis. And again, back to this idea of a viral toxin. So it's not just the blocking the virus and the infection by the virus, but, we have a protein. And fascinatingly, I know we're over time, but we've actually shown that SARS-CoV-2 spike protein does exactly the same thing.
Okay.
So when we publish that paper,
yeah, right. Come back. So Janet, can I just show this movie and you narrate it, right?
Okay.
So here it's a movie.
Yeah. So the yellow is the greasy part. David says yellow looks like butter.
Okay. And the two
NS1.
colors are green are the two NS1s.
Subunits.
Okay. So I'll start it.
Okay. Okay. So there's the dimer. And this is the hydrophobic bit. And this interacts with the membrane. This has got to do with replication, and we're remodeling the membrane. Here's the 2B7 Fab fragments. So, we can't get to the membrane. And I think that's it.
Yeah. Yeah. Those two blue parts hanging down are the antibody piece that that blocks it from getting there.
So, Eva,
and you're practicing your Oscar acceptance speech now.
Eva, I want to know if you can select viruses that are resistant to this monoclonal.
We haven't tried, but
I'll bet no.
Now that actually gets to another really interesting story, which is that the viral dissemination paper that has led us to also a re-collaboration with Richard Kuhn's group, because we found through various ways that NS1 actually interacts with the virion also. And that we think that the NS1 in a way could almost be acting like an additional receptor for the virion by binding to cells and then binding to virion. And Richard has one mutant that can block that interaction. And so we've done all this work as well, where NS1 even, so it's not a structural protein, and we actually have another paper that came out recently which gets at this as well, where antibodies to NS1 can actually kind of neutralize at high level the virion, because there's low levels of NS1 on the virion surface as well. So it's kind of like this whole other layer of complexity, which is super interesting.
But where the monoclonal binds, you can make probably single amino acid changes that block binding. Then you could introduce those into virus and see if that virus replicates at all, right? And if it does, would it be resistant?
And we've actually looked at where, because it depends on where the contact sites in relation to, because again, the NS1 is so multifunctional that there's, we have to be careful about all of that once it's in the virus. But we are making infectious clones with different mutants and could follow this up, but we don't have the answers yet.
So I have a question about the hexamer that's secreted. Do you think it's functioning mostly to mow down the glycocalyx or other things, or what?
It has to be internalized to have that effect, right? It doesn't have the enzyme activity.
And you mean how does NS1? What you're saying? How does it work with the glycocalyx or the
Well, I'm just trying to figure out what the function of the secreted hexamer is.
Yeah. Okay. Oh, yeah. So the secreted hexamer, well, that interacts, I don't know what format, but again, Michael Diamond's group had shown very nicely the interaction with complement proteins and blockade of complement pathway, which is, of course, extracellular. At least interactions with it. So there's like a whole, but I don't think that they did work on structurally what's involved. We know that, again, we have another set of data which has to do with the specificity of the different NS1s for different endothelial cell surfaces that we believe is mediated by glycan binding. And then, we also know that there's a separate internalization. Aside from the binding, there's an internalization step, which is required for NS1 to be able to trigger that pathway that leads to destruction of the endothelial glycocalyx.
Okay.
And we think that that's probably mediated by a proteinaceous receptor of some sort that we're trying to identify.
None of these are unique to the hexamer form.
No.
They're right.
We think that the hexamer, and that actually was another whole project that we had envisioned, with kind of stabilizing the hexamer, or stabilizing the dimer, and then looking for functional attributes, et cetera. And that's another project.
But I think actually it needs to be dynamic. It needs to be able to interact with a membrane and then travel around in the blood, or outside the cell. So this is just a cute way to do that.
Yeah, one thing I was thinking is the hexamer is just kind of the carpool. It's to get the dimer to places where the dimer actually does the work, and the hexamer is stuck together because
or the beach ball analogy,
or the beach ball.
I'm looking at Eva's Harris. Eva's beach.
So here you go, mRNA vaccine against NS1. We know they're great.
Right.
That should be it, right? And maybe you hit multiple, I think you were talking in the paper about designing an NS1 that would hit multiple flavies, because some are less well inhibited than others, as you said, and you engineer it. mRNAs.
It works very well. It's pretty impressive.
Yeah. So, anything we missed that you want to touch on before we say goodbye?
I just have to say that I spent a little while yesterday spinning this sucker around in the protein database. And I'm fascinated by the interlocking nature. You touched on this, the interlocking nature of the beta role between the two subunits. I mean, it's crazy. It's hard to imagine that being put together without like a chaperone or something like that.
Well, you figure that there's probably multiple copies being translated and translocated, and they get cleaved at the amino terminus pretty quickly by, I think that's
that is an amino
site.
terminal domain, right? Okay.
That's the amino terminus, yeah. And then they find each other. And
that could even see the dimerization, that interaction.
Yeah.
Well, it's a beautiful molecule. Very cool.
So
a Renaissance molecule.
Thank you.
A Renaissance molecule as well. Does everything.
Janet, any last words before we go? Janet? You're good?
No, thank you very much.
This has been incredible.
It's been great talking to you.
It's a really, like I said, in my lab and Janet's, it's a, it takes a village, several villages.
Yeah.
Well, when you when you get some SARS-CoV-2 or anything else, let us know. Come back.
Yes. Okay.
Okay. So from the University of Michigan, Janet Smith, thank you for joining us. From University of California, Berkeley, Eva Harris. Good to see you again. Thank you.
Yep. Great. Thanks.
Bye-bye.
Thanks.
Bye. Bye-bye.
Bye. I'm just stunned that the antibody is hitting this, and they just screened it by binding it. Well, I guess she has other monoclonals too, so.
Yeah, but I mean, they just happened to develop this monoclonal in this ongoing project that they've got, and it does this.
It's very nice. I think it has therapeutic promise too, therapeutic and prophylactic for sure. That's pretty cool.
Yeah, the other paper talks about the fact that because they're mouse monoclonals, they need to be able to be humanized.
Right.
And the monoclonal that's in the other paper has enough sequence character to it that they think that can happen, and probably that'll be
since the Fab fragment of that is the active part, you don't need the constant region. I mean, it seems like you ought to be able to humanize that pretty easily.
Right.
Or just make a nanobody. Maybe that would work.
Make a nanobody.
Right.
Right. The little blob, and that should stick on there and prevent it from hitting the membrane. Maybe that'd be enough. And then
they need to call up the llamas.
They need to call up the camelids. Yeah. Anyway, very interesting. Thank you for organizing that, Kathy.
Oh, my pleasure. As I said, when I heard Janet give that seminar last fall, it was just like, we have to have this.
Yeah.
We need something besides coronavirus. And this is really something to do.
This is cool. And they were cool. They were excellent guests. Really great.
I did think of one. There was one connection to coronaviruses. Did you guys pick it up?
What was that?
Cathepsin-L.
Oh, right. Yeah.
So I am going to paste in here the link to the protein database file. Okay. That I would really, listeners who really want to get into this, I would recommend checking this out, because if you just click on this link, the protein database, for those who might not be familiar with this, is a place where structures live. And it's now so sophisticated that you can just go to this link and you can get up a three-dimensional model and you can make it full screen and you can use your cursor to spin it around and play with it. And it's fascinating. And this is in particular, since we just talked about it for an hour, this is a good protein to cut your teeth on because it's not too complicated. And you can see a lot of the stuff we're talking about. It's really fun.
Yeah.
All right. Before we do a couple of email, I just wanted to point out, because many people have asked about this, Pfizer and BioNTech have started a clinical trial to test their mRNA vaccine in pregnant women. First participants have been dosed in this phase 2-3 study. Safety, tolerability, and immunogenicity, healthy pregnant women 18 years of age and older. So many people have been asking about this, so that's good. And they've already done, developmental and reproductive toxicity study with the vaccine in animals. No evidence of fertility or reproductive toxicity. Many people, there was this bogus syncytian homology story running around the internet. People are worried about a few amino acid homology between syncytian, which is part of the placenta, and the spike would be a problem. And so this shows it's not an issue, but we didn't think it would be anyway. So this is great. Couple of months, I presume, we'll have the results of that. And
now, are pregnant women being told not to get the vaccine right now?
Yeah, you can't get vaccinated. It hasn't been tested in them.
Okay.
Well, you're being told to consult with your physician.
Yes.
Right.
Exactly.
Which is being told not to. All right.
Yeah.
Well, it depends how you and your physician evaluate it. I know of pregnant women who are getting the vaccine.
Certainly there were some who got pregnant during the phase 2-3, right? So, but that's very few people. Yeah. So some pregnant women are getting vaccinated, huh? They're taking a chance, I guess.
I think so. Yeah.
It's probably okay, but you really should try it. But this is good.
It is very important they're doing this study.
That's
very good.
How many people here? Let's see.
4,000.
4,000. Good. Very good.
It's placebo controlled, so presumably. All right. Well, we don't know, but it'd probably be 1-to-1.
What are the endpoints? Obviously safety, but are we looking at immunogenicity or are we actually looking at efficacy?
It's an efficacy trial. Efficacy.
Okay.
Safety and efficacy.
Okay.
Prevention of COVID-19.
All right.
So, it'll take a couple of months to get this. And of course, today, as we are recording, the FDA is hearing the data from J&J about their vaccine. And so I'm betting that by the weekend, we'll have an EUA.
I'm expecting they're going to,
like right after the meeting today, right? Probably
shortly after.
So that's good. They apparently have 100 million doses in their pockets. Good stuff. Get them out there. And next Tuesday, we'll chat about it.
Yes.
On TWiV. Yes. Let's see. We have a guest on Tuesday. Who is it? It's Janet Iwasa. More virus structures. That'll be fun to look at that. Anyway, so stay tuned for that. And let's do a couple of emails.
Just before you do that, I looked up the clinical trial. It looks like they're trying 2 doses.
Cool. 2 doses meaning a dose and a booster, right? Not 2 different amounts, right?
Oh, yes. I guess that is what it means. Yeah. Okay.
All right. The first email comes to us from Ginger Campbell, who, of course, was on TWiV this past week. Based on the feedback I've received so far, I need to ask you to read this apology during your next episode. So I assume people are emailing Ginger because I've gotten a lot of emails which are very grateful for the episode. And,
it was great.
No complaints, but apparently Ginger got some. So I want to apologize to any listeners who feel that I disparaged or belittled the role of nurses and nurse practitioners. I acknowledge that physicians can also be the source of bad information. That is why I constantly encourage all my colleagues to listen to TWiV for accurate information on SARS-CoV-2. Obviously, both nurse practitioners and nurses are essential members of any palliative care team, but I was trying to emphasize that since palliative care is an interdisciplinary approach to caring for patients with life-threatening illness, it is important that people look for programs that include both physicians and nurses with specialized training in the field. There you go. I think she had said a nurse told her about a microchip or something like that, and that.
Yeah, I don't recall nurses or nurse practitioners being disparaged, but if somebody felt disparaged, it was unintentional.
Yeah. And let me just take the next one from Taha, who sent us a preprint or a paper. Said, this is our paper. Specific thanks to you in This Week in Virology. So the paper is called Nanoluciferase Complementation-Based Bioreporter Reveals the Importance of End-Linked Glycosylation of SARS-CoV-2 for Viral Entry. And so Taha Azad is the first author. And if you go all the way down to the acknowledgments, it's, thanks everyone. And then it says, thanks to This Week in Virology for inspiration.
Oh,
Yeah,
Isn't that cool?
Nice.
that's nice.
Very nice. I'm surprised, frankly, that the journal let them keep it. But I guess they tend to not mess with acknowledgments, right? They kind of, sometimes they reword them.
Yeah, let you do what you want. Yeah.
That you do what you
Isn't
want.
that nice? That's great.
Yeah.
cool.
Alan, can you take the next one?
Sure. Joel writes, wonderful educators. I have long heard that the process of creating the annual influenza vaccines involves a lot of time and lots of chicken eggs, with the upshot being a significant lead time between identifying the target strains for the season and actually having a vaccine available. Now that we're living in the future with mRNA vaccines in the toolbox, would this technology allow us to more quickly and routinely produce the quantities of multivalent influenza shots we need? And would that responsiveness be beneficial when surveillance indicates a particularly worrisome strain with little lead time? My question is mostly about time to produce. I understand that the cold storage and longevity of today's mRNA vaccines present their own complexities that might make routine annual use less practical. Yet the ability to respond quickly to concerning influenza strains when needed would seem very powerful. Thank you for being a constant source of top quality information throughout these challenging times. And Joel's in Denver. So there are recombinant flu vaccines in use and using various systems. They're not all produced in eggs. In fact, the whole industry is kind of moving away from that platform to cell culture based. And Vincent, you probably have more of a finger on what's going on in this technology, right?
Well, as it is, you start in, they pick the strains in January. We've had a couple of TWiVs, and by August, you could start putting them in arms, and it's a well-oiled procedure. Yes, most of the vaccines are still produced in eggs because it's very easy to do. As Peter Palese has told me, it's much easier to do that than to grow 100,000 liters of cells. Some of them are made in insect cells, which are easier to produce in quantity. But yes, we are moving, I think, away from eggs. There is a plant-based HA vaccine that is in late clinical trials, actually, and that's very inexpensive. And, you can make a new one very quickly. And I'm sure mRNA vaccines are going to be tested because it's such a successful platform. So, I mean, the turnaround it works, from January to August. That's when you start immunizing the old folk in CVSs and Rite Aids and so forth. So
it seems to me that there's sorry, Kathy, go ahead.
No, I was just going to say, I just put in 2 links. There's one describing how Moderna's already developing mRNA-based vaccines for flu, and another 2020 article in Vaccines entitled mRNA Vaccines Encoding the HA Protein of Influenza A, H1N1, delivered by nanoparticles and protection in mice. So, and I think that we, even in talking to Kizzmekia, I think that we got the idea that people were thinking about mRNA vaccines for influenza way back before it was even on our radar with respect to
everybody who develops a vaccine platform, I think, thinks about using it for flu vaccines.
Right.
The influenza, I mean, the Moderna has a page that shows their products in the pipeline.
Mm-hmm.
And they have something they call flu vaccine, or 3 different products they call flu vaccine that they say are in preclinical development. And then they have an influenza H7N9 vaccine that has been through a phase 1 trial. But my guess is, all of this is going to get, all of the mRNA technology is going to get ramped up as a result of what's happened. I mean, it seems to me there's 2 advantages here. One is you don't have to make virus. And the other, but as you've already pointed out, for a lot of the flu vaccines, you no longer have to make virus anyway. But now you don't even have to make the protein outside of the virus and purify it or anything. Theoretically, you can just inject the messenger RNA. And do you see any reason why that shouldn't work, the hemagglutinin by itself? I mean, if you can make it in insect cells,
plants, that's all they have is the HA,
Yeah, you ought to be able to do it from messenger RNA, inject it into a person.
I remember the other thing that's going to evolve are the universal flu vaccines, right? So that's a whole different story. Some of those are actually egg-based, but others are just HA, so that might use it as well.
I keep thinking that decades from now, they're going to look back on this and they're going to see it as on the one hand, a tragedy, and on the other hand, a scientific revolution.
Yep, I agree.
That's amazing.
I agree.
So the flu vaccines are called mRNA 1010, 1020, and 1030. And so, Vincent, I don't know how you're going to keep all those numbers straight, but and then against HIV, they'll be called mRNA 1644 and 1574. And they're also working on one for Nipah that's mRNA 1215.
Sure.
Is that the length? Are those the amino acid lengths of the proteins, maybe?
I don't know.
At this point, Moderna should be able to afford to hire somebody to come up with better product names. I'm pretty sure they can budget that.
And then they have, parainfluenza type 3. It's mRNA 1653. So,
I do like the mRNA 1273 because now, as I said before, I know the length of spike because I really can't remember the lengths of proteins, but that one. And that's pretty long because some other immunogens were 300-400 amino acids typically, and that's big. So,
but people should bear in mind that the mRNA vaccines, they have a lot of advantages, but they're not magic. So just switching to that platform is not suddenly going to give us a 95% effective flu vaccine.
It's not guaranteed to work, but if they hadn't been rolled out so quickly in this pandemic, we would probably be taking years to get them out there, right? So
it will actually be interesting to see how well this translates to other pathogens. Whether we got lucky. Okay, or whether this is really is pretty broadly applicable.
Well, and we have examples of other vaccine platforms developed in a similar timescale. In this case, we've got J&J, which is being discussed today. We've got AstraZeneca. We've got the Sinovac vaccine. We have the Sputnik vaccine. So there these other platforms can also be done fairly rapidly.
Yeah, as a matter of fact, people always say, oh, the mRNA thing is really easy. And I don't see how it's any more difficult, really, to make a recombinant virus once you have the sequence.
No, right.
I have to say that I get a lot of emails from people who object to the fact that even though cells are not used to produce the mRNA vaccines, a long time ago, they did put the mRNA into cells to show that the protein is made. And do you know what kind of cells they used?
HeLa cells. How about that? No?
HEK 293. And those, of course, are made from an aborted fetus in 1973. And there's people who simply will not touch it because of that. And it's your choice but those are the facts. We're not hiding anything. They are. And I went into the Pfizer and Moderna FDA document papers, actually, the phase 1 papers. We checked that the mRNA makes protein by putting it into HEK cells. So, even though it's not used in the production, if you object to those and you object to them checking protein production, then you're right, I suppose. Kathy, can you take the next one, please?
Martha writes, I'm so thankful for vaccines. My daughter ended up hospitalized with rotavirus while the RotaShield was off the market. She got really sick, lethargic, hypoglycemia, hypotension, and needed multiple fluid boluses. She scared me, and thankfully she recovered. I wished she could have had the vaccine. As a little girl, I had a family friend who died at age 7 from Reye's syndrome following the chickenpox. I remember thinking that I would die when I turned 7 from Reye's. Clearly, I was too young to understand. As a teenager in the 80s, I took care of a woman in an iron lung from polio. She spent her time in a rocking bed or the iron lung, a difficult life following polio infection and severe disease. I've seen healthy kids lose their life or suffer severe sequelae, missing limbs, brain injury, deafness, or more from meningitis. And I've cared for immunosuppressed transplant patients ill with vaccine-preventable illnesses who desperately needed strong herd immunity. As a 1A healthcare worker, I am happily vaccinated against SARS-CoV-2, Pfizer 2 times. The vaccination was easier than the shingles vaccine for me. I love a good immune response that tells me my immune system is responding. I love the TWiV podcast and recommend your podcast often. Go science, Martha. Thank you, Martha. All of those are great stories that are the kinds of things that really help to convince the vaccine hesitant.
Absolutely. Absolutely. Rich?
Noel writes, I feel like I know the answer to this, but would love a clarification from the experts being you, of course. I have MS and asthma, so I have opted to step out of the workforce for the time being. My husband is a firefighter in a major city, and we depend on his income. It is understood by both of us that his is the risk we must live with. He uses every precaution recommended and has been as careful as possible while working. He always masks when running our errands and doing our shopping and is great about cleaning, etc. He has had both doses of the Moderna vaccine. I have not, due to severe allergies, anaphylaxis, and multiple medications. We have no outside guests and haven't even seen our kids or grandkids for over a year now. So imagine my surprise when I see a confirmation from his work for an upcoming flight.
Oops.
He is scheduled to take an out-of-state trip with several other firefighters for a conference of some kind. I know they will all have separate rooms, etc., but this seems like a risk we should probably not take, especially until I have been vaccinated. If he does end up going on this trip, how do we handle his return? I feel like he needs to test when he gets back and quarantine out of the home for 7 days. Is this the safest route for me? We have an extra guest bedroom, bathroom, but I don't know how we would handle meal times, etc. if he were in the home. I just need some sound facts to pass along to the therapist/moderator that I am no doubt going to have to hire and involve in this upcoming battle. Thank all of you for being there throughout these interesting times. I really respect your opinions, and I'm constantly telling friends and family all of the things that my scientists have taught me. You're all great. Thanks again, Noel. Tough one. So what's the risk? The risk is that, so as we've said so often before, although the 2 doses of the vaccine will likely protect you against getting serious disease, we don't know whether or not you might yet be susceptible to, shall we say, an asymptomatic infection. So there is there are some data that we have discussed before that says yes, vaccinated or immune people can get asymptomatic infections, and they may have a reduced viral load. That's, at this point, almost anecdote, okay, that we don't have any, lots of data in that regard. So that's the risk, that he goes on this trip, acquires an asymptomatic infection, and brings home some virus at some level. So I agree that the absolute safest thing you could do is for him to get tested and perhaps to quarantine out of the home for 10 days. The 10 days, I think, is what the CDC would do. That seems extreme, but if you want to be absolutely certain, that's if your answer is that what's the safest thing I can do, that's it. And all of the issues that you talk about trying to quarantine in a separate bedroom are, all legitimate issues. Trying to quarantine somebody in the same house is difficult. The other thing that comes to mind is that, if you're unvaccinated, when are you actually safe? Okay. To resume something like a normal life. Normal even including, your husband resuming some sort of normal activities.
Well, I think when we've got most people vaccinated, we've cut down, cases, then yes, you can. And she who has MS will not get vaccinated, but if she gets sick, she could get monoclonals. Right.
Okay.
So I think that's what we have to do, because you can't have people who can't be vaccinated not have their normal lives, right?
Right. I think there are a couple of things here that I would point out. First of all, it matters a lot what state he's going to be traveling from and to. Most, if not, maybe not all states, but many states have travel restrictions in place, and they're keeping them in place. And I don't really agree with all of them. I certainly think a lot of what Massachusetts has done is kind of counterproductive and stupid, but it is a requirement. For example, if I were to fly to any other state besides, I think Hawaii is the only one that's not designated plague zone for Bay Staters. On my return, I would have to present a negative SARS-CoV-2 test taken within the previous 48 hours in order to be allowed back into the state of Massachusetts without having to quarantine under Department of Health supervision. So that procedure may already be legally enforced when he comes back. And if that's the case, then he should obviously follow it. If that's not the case, then as Rich said, the absolute safest thing would be to plan on a quarantine of up to 10 days. But if he gets tested right when he comes back, test results are now typically turning around within 24 hours or so. So if he just stays an extra night in a hotel and gets room service in your local area while waiting for his test results to come back, and then they come back negative, then you should be okay from there. If they come back positive, then you would go through the contact tracing and isolation procedures that are standard for that.
The problem is that if he's infected on the last day on his trip, the incubation period could be 2 to 14 days before, so it could be longer before he's PCR positive. So
yes,
it's a problem.
It could be.
Right.
And in terms of what people who can't be vaccinated will do, first question to Noelle would be has she talked to her doctor about, are there circumstances under which she could be vaccinated? Because that would be the first choice. And bearing in mind that quite likely we're going to have more than just Pfizer and Moderna mRNA vaccines on the market soon. And that's one of the big selling points, I think, in addition to the added supply and the single dose aspect and the refrigerator storage, the J&J vaccine has the advantage that it's a totally different platform. So the anaphylaxis contraindication may or may not apply. Might be something to look into when that's approved. But for people who really can't be vaccinated, I think we're going to have to figure out at some point, what's the acceptable risk? And a lot of people in public health are throwing around the number, 100 deaths a day is what we tolerate for flu. And the public seems to be comfortable with that. And so when SARS-CoV-2, when COVID-19 deaths get below 100 a day, that may be the point where we say, okay, you know, we as a society are comfortable accepting the risk, and some people are going to end up in the hospital. And as Vincent said, you know, there are now much, much better therapies available than were the case, and better treatment available. So, you know, hopefully not going to be an issue, but that's there.
So I have a couple of comments. One is that, kind of going along with what Alan said about depending on where he's going, I question how much more risk there is in doing that versus him going out on errands and doing the shopping.
And doing his job.
And
Good point.
So,
Good point.
You know, if you're already comfortable with that, and if he were to come back and quarantine, you know, get tested right away and quarantine and get tested again in, you know, 5 or 6 days, when, you know, I'm not sure about the timing on all of that. So that's one thing, but you know, she does say until she can get immunized, and I would also agree with Alan that, you know, she should talk to her physician. I don't know the risks for MS, but the anaphylaxis, from what I understand, has been dealt with it's something like 10 cases out of 2 million or 10 cases out of 4 million.
The rate is about one in 100,000, and no deaths. Everybody has been treatable,
right,
on site,
right. So
yeah, there's a. In fact, when you go and you get your shot, you have to wait. You're detained in a waiting area for at least 15 minutes. The place I went, they had a big clock on the wall. They said you can't leave until this time. And there's I think we had a doctor and a paramedic sitting in the room, and EpiPens available. And so if somebody had gone into anaphylaxis, they were going to get an EpiPen, and they were just, you know, going to be transported from there.
Right.
I think that's a really good point, Kathy. Is the trip actually any more dangerous than being a firefighter?
Right.
And only they can evaluate that. Right. That's a good point.
Maybe he'll have less risk when he goes away. Yeah. Good point.
Yeah.
And also, just based on most recent stuff, that supposedly the cold storage for the Pfizer vaccine is going to be mitigated somewhat. That and somebody wrote to me and said there's going to be a huge glut of -80 freezers available. and that's good, because I know of some labs that need to buy a -80 and they cannot get one. So, yeah.
I just want to read one more email. This is a physical card that I received here.
Oh.
And LOVE, and it's handwritten. And these are little stickers stuck on here, cutouts, which are done by the daughter. P.S. My daughter is the card designer, right there, right? Nice. So this is from Leslie, who writes, "Dear Vincent and the whole TWiV family, wishing you all a sweet, peaceful, and healthy V-Day, filled with love and affection. Thank you for all you do to keep us informed and up to date on all things SARS-CoV-2, COVID-19. Your thoughtful discussions have kept me going through this past frightening year. I sing your praises at every opportunity. Keep on keeping on. Love, Leslie." So that was mailed to me here at Columbia. Thank you, Leslie. Some people do write letters still these days. I always like when they're nice.
I got a postcard that was postmarked November 30th, and I got it on February 19th.
Why so long?
The post
USPS.
office, the post office had a really bad patch right around the time they were being sabotaged for the election. Oh, sorry, I didn't mean to get political, but
yeah, and postcards are always lower
postcards are
priority.
last priority, and Christmas also slammed them because FedEx and UPS were already overwhelmed, and they just stopped taking packages. But the Postal Service can't stop taking packages. So I actually had some Christmas packages I sent out to people that took a month to get to them.
Yes.
Where did it come from, Kathy?
Georgia.
Wow. At least it didn't come from Lansing.
No, right.
Which is one of the few towns I know immediately in Michigan. Milwaukee. No, that's Wisconsin. Lansing and Ann Arbor. What else? What is another town in Michigan that I might know?
Detroit.
Oh my gosh. How could I forget Detroit?
Flint.
Yeah. Yeah.
Flint. Okay.
Yeah.
Flint is, yeah. Speaking of politics, Alan, Tony Fauci the other day said because of politics, we had not a good response to the pandemic. He came out and said it. And I thought, we said that 10 months ago, right?
Well, he wasn't allowed to say that.
He wasn't allowed to say it. But when we said it 10 months ago, people said you're being political. But it was the truth.
Yeah.
Let's do some picks. Alan Dove, what do you have for us?
I have so I'm linking to a BBC article on this, but it's you can also find from there the Royal Photographic Society site, and they have a Science Photographer of the Year competition, and so they have just announced their winners, and they're pretty cool. Some very, very good images. And you can go ahead and scroll through and take a look at those.
Yeah, very nice.
Some of them are pretty depressing, but they're all they all make you stop and think.
Yeah, the spherical lens inside a pattern tube.
Yes.
Wow, very nice.
Poor whale.
Excellent.
Poor whale.
Yeah.
I do like the polar bear on the ice floe. That's just great.
It's a stunning shot. Yeah.
It's really cool. You think the whale will get out of that? Well, maybe the photographer would help, right?
I think that may have been done as part of an effort, like maybe they were there to try and help.
Yeah.
I like to think that.
Hopefully, yeah.
Yeah.
Well, Dixon would like this, but of course he's not going to ever see it. So,
well, we can send them to him because I think I'm going to send him my picks too. So,
Rich, what do you have?
This is a little strange. This comes under the heading of how a nerd keeps house. There's this product that I was turned on to some time ago called Barkeeper's Friend, which is it's a cleanser, a powdered cleanser that I was introduced to for the purpose of cleaning stainless steel, in particular a stainless steel cooktop, because it's a marvelous cleaner that doesn't clean you at the same time as would oven cleaner, and is not abrasive, really. Well, it is abrasive, but not sufficiently abrasive so that you scratch your stainless steel.
Right.
And so, this is the nerd part. I use this stuff, and it just blows my mind because it's just great, and it's great on grout.
It's oxalic acid.
Steel. Oxalic acid is that's right. And if you look into their Wikipedia site, of course, my first go-to, you find this stuff has been made since the late 1800s, and they tell a story about some guy who was a chemist who was cooking his dinner, and after he cooked some rhubarb, looked back at the pan, and the pan was cleaner than he'd ever seen it. And he said, being a chemist, he said, how can that be? And he figured it out. Rhubarb is very rich in oxalic acid. So its properties that are relevant is that it's acidic. It's also got mild reducing properties. So it's great on copper pans as well. And I looked it up. I looked up the materials data safety sheet, of course. It's got a little bit of feldspar in it, which I think is where most of the abrasive stuff comes from, mildly abrasive stuff.
I've been using BKF since graduate school. Great stuff. Yeah.
Great stuff.
Wow. It's the simplest it's the simplest carboxylic acid. Did you know that?
No, but I get it now that you mention it, because of course I've looked up the structure.
Yeah, it's beautiful structure, and it's used by some beekeepers as a miticide against the varroa mite. They sprinkle it on their honeybees.
Oxalic acid is also the active ingredient in some teak cleaners. If you have a boat that's got teak on it, you need to deal with that.
Well, if we're going to get on a cleaning product arc, I found somewhere I had a pan that couldn't get clean, and I read that you take baking soda and vinegar, and you throw it and let it sit for a few hours, and boom, it comes off without even scraping it. Right? I was amazed because we have a lot of that stuff around. My wife put.
And by the way, another little science in the kitchen thing. If you ever get sugar burned onto a pan,
yeah,
maybe you were trying to make caramels and it failed. My first effort was with a Dremel tool, which I do not recommend.
Yeah, that's not
but
a good
then
idea.
I found out that what you can do is boil hydrogen peroxide in the pan. Because it has a higher boiling point than water, and it will melt off the sugar as it boils.
Oh,
Cool.
BD.
Very good.
Anyway, that's it. We're just doing picks of the week, and we're digressing.
Yeah, I typically do just some dishwasher powdered, detergent overnight in a pan, or sometimes if I think it's going to need it, I'll boil it on the stove. But yeah,
Doesn't work for the burnt sugar.
I think this was a dessert pan. Yeah, and but the baking soda vinegar works. Yep.
That's good.
Rich, that was you. Kathy, what do you have? What do you have for us, Kathy?
I have a rich style nested set of picks. So first was the Perseverance parachute that carried a secret message. So if you haven't seen this, I linked to the Washington Post article where the parachute says something that's evidently found all over JPL from Teddy Roosevelt. And it's in binary, and it says, I can't read it here and I don't have it. It's rats. Come on. Why don't you guys just say it for me?
Dare mighty things.
Dare mighty things.
Yes. Okay. Dare mighty things. And then it has the coordinates of a spot that's just outside the door of JPL. And so this is, I was trying to figure out what the code is, and it's actually like A is the first letter. And so the code for A is 1 in binary, and D is the fourth letter. So the code for D is 4 in binary, because there's also an ASCII binary code, and I could not make that work. So I had to call up my brother, and he said, no, it's just the numbers of where they are in the alphabet. And so then that led me to another thing, or in that article, it said that there's also pictures of all 5 of the rovers that have been to Mars on the outside of the thing. And so I found a picture of that. And then there's also in that same set of pictures, it doesn't say where it is, but I assume it's somewhere in Perseverance. And it shows a DNA helix, a dinosaur, I think a fern in the background, silhouettes of a man and a woman, a rocket, and so forth. So a variety of things, Easter eggs that are associated with Perseverance. And then there's also 11 million Earthlings' names in it on 3 microchips, and essays of 155 finalists in the Name the Rover essay contest. And then there's a link where you can sign up to send your name on a future Mars mission, and already almost 8.7 million people have signed up for the next mission, which is July 2026. And I am one of them. So
yeah, I need to do that.
Wow. I figured someone was going to pick this. Yeah, when I saw this.
Yeah.
That's pretty cool.
Very cool.
All right. My pick is a new effort by Google. Google has funded a COVID database to track variants and immunity. So I have a Nature article by Amy Maxman that describes it, an international database. This will have much more data than any of the tracking databases that you're used to. They're going to have, up to 40 variables, like when the symptoms began, when the positive test, travel history, and so forth. So the actual site is global.health. It's funded by Google. And so far, they have 5 million anonymized cases from 100 countries, and you can go in and explore all the data. And I think that's great.
Cool.
I'm going to send this link to my son, see what he can tell me about this.
yeah.
Interesting.
Yes, so Google has been doing a lot behind the scenes. They preferred to maintain a low profile last year so as not to be called out on Twitter by a certain individual. Of course, that individual no longer has a Twitter account. So perhaps now they're becoming more, they have a COVID response team that they've been doing behind, and I've been meeting on their calls every other Sunday. And it's quite impressive. They're doing it at their expense, and they're doing a lot of amazing things. And I guess this is one of them. So they just announced this.
Well,
so
they have the money. Might as well do something good with it. That's good that they're putting it to use.
They do. And we have 2 listener picks today. One is from Matthew. I wanted to share the COVID memorial that New York's Metropolitan Transportation Authority commissioned in honor of its fallen workers, entitled Travels Far. As you might imagine, MTA's frontline workers paid a tremendous price throughout the pandemic. What is also instantly evident in this memorial is the racial and ethnic disparities that were exploited by the pandemic. So this is a lovely memorial where... it's to the lost colleagues, pictures of everybody, and you can click on them and, see how long they lived, what they did. Bus operators.
A lot of people. A lot of people.
Tons of people, unfortunately. These are people that ran the buses, the trains, worked in the stations. Really unfortunate. And
these are
see
people who kept the city running. I mean, if you've been to New York, you know how important the MTA is. The place will not function without these folks. Wow.
Yeah, a lot of
people. Is this going to be, are they going to have something besides a web page for this, like a mural or something, maybe in the subway?
I'm sure they will.
That would be good.
At some point, yeah. Somewhere in New York, they'll put up a memorial, I think.
Yeah.
That would be really good. Yep.
And as I quickly scan through this, I think I'm safe in saying that these are predominantly people of color and other minorities.
Yep. Ethnic disparities. Yep. Racial and ethnic. Yeah, absolutely. Absolutely. So thank you, Matthew. And then Lori writes, I love this show. Every time a new episode drops, I can't wait to go for a run. Listening is how I motivate myself to run in the winter drear. I just bought a TWiV hoodie. Love it. All right, so Lori couches a question in with her pick, which is very clever. I've heard of several cases in which both members of a couple die from COVID, where the couples are in their 40s or 50s. The couple I know had no comorbidities. What could cause this? Or is it just the randomness of this virus? A couple meaning, they've married or are living together, but they're unrelated. So it's probably just random, because there are interesting examples of families where, say, one child will get very sick and the other will not. And that could be a genetic polymorphism. But I think it's just random. They both, unfortunately, no comorbidities, but they probably had something that we don't know about. Some polymorphism that makes them susceptible.
Could be microbiome related.
Could be microbiome related. Yes. If anyone tells you to change this show, don't listen to them. Keep it exactly the way it is. Many people tell us to change the show. Always something. I want to change this, change that. Yeah, thank you. We will do our best. And her pick is great. There's physics involved, and it's fun. It's a site called Iceberger. Draw an iceberg and see how it will float. And so there's this thing they give you, and you can use your mouse to draw an iceberg. And it says that icebergs are less dense than water, so they always float with about 10% of their mass above the water. But which way up? An iceberg wouldn't float exactly like, on this page, reality. Its three-dimensional distribution of mass and its relative density compared to the water are both significant factors that only approximate it here. So you can draw a bunch of them and see if they float.
This is so fun.
Isn't that cool?
This is awesome.
It's very cool. And you can keep drawing them over and over again. You can actually just draw a line. It'll finish it for you. And
gosh, this is great.
You see if it floats.
Okay, Rich, I know what you're doing with the grandkids.
I'm going to be here for hours.
Yeah, I just sent the link to him.
It's very cool. Thank you, Lori. It's awesome. And for that, you get a question answered, which was not really answered because we don't know. Anyway, that is TWiV 725, microbe.tv for the show notes, questions and comments, TWiV@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit, Emeritus Professor, University of Gainesville, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.